Ribosomes. Ribosomes are these little dots that you're looking at there. Ribosomes, which are little protein factories, a fancier way of saying that is that they're the site of protein synthesis. So synthesis means to make, so it's where proteins are made. The actual name of the process of making proteins is called translation, and we'll talk more about that when we get to genetics. Now, ribosomes can be found in a couple of different places inside of the cell. This first picture that we're looking at here, they're attached to the rough ER. They're responsible for the rough appearance of the rough ER. But you may, oh, here they are again. There's the endoplasmic reticulum, and it looks like a gazillion little ribosomes attached to it. They may also be found free floating in the cytoplasm. So this isn't the surface of the moon. This is your cytoplasm, and those are ribosomes. Now, if you notice, you can kind of see some texture there, some different shape to them other than just a little circle, a little blob. And this is why it is actually a two-part structure. And we will see when we get to genetics. Yes, we'll talk about the making of proteins in genetics. That's where it happens. That there's actually a type of genetic material called the ribosomal RNA that is a part of the ribosome structure. And then the message, the information comes from the nucleus, the DNA in the nucleus, and it will be brought here to assemble protein. So that means the linking together of amino acids with what are called peptide bonds to form the proteins. Now, what are you going to do with them? Well, this is what's going to happen to them. The ribosomes will produce the proteins, and then these proteins will be sent to this structure called the Golgi apparatus, which can also be called the Golgi body or simply just the Golgi. And this is where the proteins are kind of sorted and repackaged and sent out where they need to go. Now, if you look at this, sometimes people will confuse the structure with the structure of the smooth ER because of the appearance. It doesn't have anything stuck to it. And the big way to tell the difference is the smooth ER, those endoplasmic reticulums, they are continuous. This, as you can see, are separate little areas that seem to be little flattened areas. And there's this kind of distinctive shape where it's kind of skinny in the middle and then it seems to get bigger at the end. And the reason that it's getting bigger at the end, what is going to be produced are these little cell membrane packages. And in those little cell membrane packages are proteins. And what we call those little packages, we call those little packages vesicles. And these vesicles, you can see a lot of them seem to be very close to this cell membrane, and this could be for exporting materials out of the cell, which is called exocytosis.